students, in the previous part we had studied about RBC, that is red blood cells or red blood corpuscles. Now the next formed element is the WBC or white blood cells. So the second part is WBC, that is white blood corpuscles, or cells. It is also known as leukocyte. Students, at some places you will also find this spelling. This is also correct. So this is the leukocyte. Now we will study WBC on the basis of similar points taken while studying RBC, such that you can study both the topics in comparison. Like there is a term used called leukopoiesis, that is formation of WBC. Now the formation of RBC was erythropoiesis and formation of WBC is leukopoiesis. Now students, we study one by one. Like first of all, its shape. WBC is irregular amoeboid in shape. Irregular amoeboid in shape, and it has one significance, that it can produce pseudopodia and squeeze out through the wall of blood capillary, and when this process takes place, then this is known as diapedesis. So students, due to the irregular amoeboid shape, it can easily squeeze out through the pores, through the wall of blood capillary, and enter into the tissue space, where it helps in engulfing the germs or fight against the germs by producing antibodies. Now after shape, we see here its number. Now RBC was about 5 millions per cubic mm of blood, but number of WBC is much less than it, that is only 4,000 to 8,000 per cubic mm of blood. Now its number is less because simply you see, a student, that WBC acts like a soldier. It has to fight against the germs, foreign substances, and so in normal condition it has a normal number, normal range of number, but when any kind of infection takes place, then its number will increase. So during the pathological test, when the blood test is performed, then the increased number of WBC indicates infectious condition. Clear, student? Now its number can be increased or decreased abnormally. So you see, that is, if there is abnormally increased number of WBC, leukemia, also termed as blood cancer, so it is abnormally increased number of WBC producing cell. WBC producing cell at the cost of RBC. Now in this case, it is also termed as blood cancer and it is fatal for the organism. Now But if its number is decreased, then this is called leukopenia. Leukopenia, that is abnormally decreased number of WBC. Decreased number of WBC. So these two terms are used for the number. Now WBC, you see, again we have to study here about site of formation. Site of formation. Now in the red bone marrow, actually student, it is produced in yellow bone marrow. There are two types of bone marrow in the long bones, red bone marrow and yellow bone marrow. WBC is produced by yellow bone marrow mainly, but in syllabus we have given red bone marrow. So we write red bone marrow here. Now in spleen, liver and lymph nodes. So it is the site of its formation. Site of destruction. And it is similar to RBC. That is, the places where RBC is destroyed, WBC is also destroyed, and further we see that WBC is destroyed mostly at lymph nodes, where tissue fluid is filtered out from the germs, and the WBC, by localizing there, fight against the germs. Mostly WBC remain present outside the blood vessel, that is in tissue fluid and in the lymph. For short duration, that is it during its flow only, it remains inside the blood vessels. Now, lifespan. It is 2 to 3 weeks. 2 to 3 weeks, the average lifespan. Now we see that monocytes get destroyed in 2 to 3 days only. So overall, this is the average lifespan of WBC. But you see, you have studied that RBCs are having 120 days. So this is the comparative study of WBC. But WBC is of different types depending upon its presence of granules in the cytoplasm or absence and other coloring factors. It is divided into major two groups, granulocyte and agranulocyte, that is granular or non-granular. So WBC is classified on the basis of, first it is granular or granulocyte. Now, as the name suggests, cytoplasm contains granules. Cytoplasm contains granules. Now, in granulocytes or any WBC, we have to find it out on the basis of types of nucleus present. In RBC, matured mammalian RBC, there was no nucleus, but WBC contains a nucleus. But it is not uniform. The WBC has lobed nucleus, that is beaded, lobed, swelled, then narrow, again narrow stripe, then swelling. So this is called lobed. Now, on the basis of this, granular part is of different types. First of all, it is neutrophil. As we have studied here, that is granular are classified on the types of coloring matter, that is staining material. While preparing their slide, while pathological observations, these different types of cells are colored and identified. Now, the first point. The different types of WBCs will be studied basically on the basis of 2 points. One, the type of nucleus, and second, its major function. Here we will also take one more point, that is the type of coloring material which suggests its name. Now, neutrophil, here it forms about 62% of WBCs. Then next part, neutrophil takes neutral dye. Neutral means it will be neither acidic nor basic. It will be neutral coloring material. Now, the most important point in neutrophil is that is it has 3 to 4 lobed nucleus. Now, what is, suppose here is the WBC, then what is the lobed nucleus? Now, this is the lobe. This is the lobe, narrow stripe, and then again lobes, and then this is the cytoplasmic part. So, this is 3 to 4 lobed nucleus. Now, it has maximum number of lobes. In ICC board exam, it was asked that identify the type of WBC which contains 4 to 6 lobed nucleus. Then it is also neutrophil, because the neutrophils have maximum number of lobes. In books it is given 3 to 4, but if 4 to 6 is given, even in that case it is neutrophil. Now, it has main function, that is it engulfs germs by phagocytosis. That is the process of engulfing germs by developing pseudopodia around it is known as phagocytosis. Like you see, suppose here is the WBC and here is the germs. Now this is the pseudopodia, so it surrounds it and then finally engulfs where it will be destroyed. So this is the neutrophil. Now, the second part of granular or granulocyte is eosinophil. Eosinophils. Now, eosinophils forms 2 to 3% of total WBCs. The main point related to it is it has bilobed nucleus. That is, the nucleus will have only 2 lobes. Like you see, suppose here is the WBC, then it has just 2 lobes. Bilobed nucleus. The next point, which is also important again, like this one, that is it takes red colored acidic dye eosine. Now, the next point related to its function, that is it produces antitoxins. Antitoxins are also kind of antibodies which are produced by WBC against the toxic substances produced by the germs when they enter into our body. So they produce antitoxin as well as, and it is related to allergy. That is, in case of allergy, the eosinophil count is increased. Clear, student? So this was the second type of WBC, granulocyte or granular WBC. Now the third type of it is basophils. Now again the name suggests that it takes basic dye, means it takes basic dye methylene blue that is blue colored basic dye called methylene blue. Now it has single large nucleus like this. Suppose here is the WBC, then here will be the single large nucleus. Now next, related to its function, it produces histamine. It produces a chemical called histamine. Wherever there will be a technical term, we must remember to underline, because the question is asked directly that name the chemical substance produced by basophil. It is histamine, and what happens due to its presence, which dilates blood capillary, and helps in diapedesis of WBC. That is, due to secretion of this histamine, the wall of blood capillary dilates, and through the pores WBC can easily squeeze out into the tissue spaces, tissue fluid, where it will fight against the germs. So we have studied the three types of granulocytes, that is neutrophil, eosinophil, and the basophil, and related to their function. Now the second type is agranular or non-granular, also known as agranulocyte. That is, cytoplasm does not contain granules. Now it has two parts mainly. One is lymphocyte. Now actually all the types of WBCs are important, but the neutrophil and lymphocytes are in larger number, therefore they perform the major functions. Lymphocyte forms about 30% of WBC. And the most important point related to it, that is, it is the smallest of WBC. Students, many times students remember this term, this word from here, that is, it is the smallest of WBC. And when the question is asked, name the smallest cell in the human body, then many times they get mistaken and write lymphocyte. But here you remember, it is the smallest of WBC, whereas the smallest cell in human body is the RBC. Clear, student? Now it produces antibody. So the main function performed by neutrophil was phagocytosis, where it engulfs the germs and destroys them, and here lymphocyte mainly produces antibody. As the name suggests, it fights against the foreign substances, the germs, kill them by changing their chemical nature, destroying their toxins and so on. Now here, that is large indistinctly lobed nucleus. So the main point is related here, that is the smallest of WBC, and it produces antibody. The second part in it is monocyte. Now it has single large nucleus again, and the most important point is that it engulfs germs. That is phagocytosis. And the most important thing is that at the site of injury, it changes to macrophages. Macro means large, phages term for phagocytosis, that is it changes to large sized cells in order to engulf the germs and foreign substances more easily. So students, these were the 5 types of WBC. Now if we study the function of WBC, so if question is asked directly to what is the function of WBC, then we can write that it provides immunity. That is resistance against diseases. Means it prevents our body from the germs, from the infectious substances, and keep us away from the diseases by killing or destroying the germs. Now, how is the function performed? There are some terms related to it, that is first phagocytosis. That is the process of engulfing germs by producing pseudopodia around it, around germs, engulfing germs around it. Now you see a student, at the site of injury, WBC squeeze out through the wall of blood capillaries, and at that place it produces pseudopodia around the germs, engulf them and destroys them. So the first process to provide immunity is the phagocytosis. Second, it produces antibody which reacts with protein of the germs or germ substance and alter their nature. In this way, the germ will become harmless. It is rendered harmless. It is destroyed. At the place of inflammation, the localization of germs take place. Localization of WBC takes place. As a result, at the place of inflammation, you see that is at that place it turns red, the temperature is increased, and in this way you see that is it helps us in providing immunity. Now, a student, diagram based question is asked sometimes. Like you see, suppose if we have this diagram. Now suppose here it is given irregular. Now here this process is shown A, WBC again here. This is the germs B, and in next part germs have entered. So question is asked like this is the RBC, platelets, WBC, this is for blood capillary. Now in this part you see the process taking place is diapedesis. That is the process of squeezing out of WBC through the wall of blood capillary. In second part, here phagocytosis is taking place. Phagocytosis is taking place. And finally, at this place, the process is intracellular digestion. Intracellular digestion. That is, the germs are engulfed and destroyed inside the WBC. So, this phenomenon is responsible for the providing process of immunity by the WBC. Clear, student? So, these were the different terms related to WBC. Okay? Students, the next component of the blood, the formed element, is the platelets, also known as thrombocyte. Now, it is sometimes also called a non-cellular component of the blood because this does not contain definite nucleus. It has no nucleus because platelet is not formed by the process of cell division. Actually, it is the fragments of some large cells, giant cells present in the bone marrow. So, here it is formed by fragmentation of some giant cells in bone marrow. So it does not contain nucleus. It does not contain nucleus. This is why it is also termed as acellular non-cellular component of the blood. Now, platelets have their number 2 lakhs to 4 lakhs per cubic mm of blood. 2 lakhs to 4 lakhs per cubic mm of blood. Now, the lifespan is 3 to 5 days. So, this is about the platelets. Now, in viral dengue, which is a kind of viral disease, viral dengue, the platelet count is decreased. Platelets count is decreased due to which blood does not clot properly and it may lead to hemorrhage. Clear, student? Now, if we study the function of platelets, now it helps in blood clotting, also known as coagulation of blood or coagulation, and prevents blood clotting or coagulation. Now it prevents excessive loss of blood from injury and prevents entry of germs through cut part. So a student, in this way it prevents us from excessive loss of tissue fluid from the body, entry of germs through the cut part, and hence prevent infections from the germs. Now, so students, we have studied the different components of blood, the plasma part, the cellular component, that is the formed element, RBC, WBC and platelets. In the next part we will study about the clotting mechanism of blood clotting, its significance, the blood group, RH factor and other topics. Clear, student? Okay.